My name is Richard Wade-Martins. I run a research group at the University of Oxford, and we're working to look for new novel targets to treat Friedrichs Ataxia. Our work is described here. It's a partnership project with Oxford, working together with Ataxia UK, Friedrieichs Ataxia Research Alliance and Pfizer to try and develop a program to find new targets to treat Friedrichs. So what we're attempting to do here is use a new technique called CRISPR-Cas9 genome engineering to identify new genes which, when altered, will elevate the protein frataxin, which is lacking in Friedrichs patients. So Friedrichs patients have a genetic mutation in their frataxin gene, which means they just don't make enough of the frataxin protein. What we'd like to do is identify and knock out genes which, when they go down, might cause the frataxin to go up. So what we've developed is an assay in which we're using this Cas9 CRISPR engineering to target genes at random in a patient cell line from an individual with Friedrichs. If we can identify genes which we knock out with this technology, as they go down, they cause the frataxin to go up, that means that that gene could be a potential target. So this poster here, work from Gabrielle and others in the laboratory, is setting this process up. First of all, it's shown in outline here. We target the genome at random with this Cas9 CRISPR technology, shown here as a scissors, which are going to cut out genes at random. And we can cut out genes and identify those which, when we've cut them out, they cause the frataxin levels to go up. And we can then, through DNA sequencing technology, identify these new cuts that we've made. And as we've cut some genes out, as they go down, we cause the frataxin to go up. So we've confirmed that the Cas9 technology works in this FRDA, this patient cell line here. We've shown that we have a methodology to fire individual cells, the capillary tube, by a technique called FACS, to identify cells in which the frataxin protein has gone up. So we can separate out on this chart here cells from a patient which have low levels of frataxin from those which have high levels of frataxin. And by identifying those high level cells, we can zoom in on our new target. And we've identified some positive controls which will show us this assay, this screen will work correctly. And we've obtained a library of vectors, a library which contains thousands and thousands of targets to individual genes. And we've shown here in this work from the lab, which we can transduce. We can put these viruses inside the patient cells. And this final panel here shows that we can then take those cells that we've manipulated with this virus, make some DNA and identify the genes that we've targeted. So, in summary, the idea is to do a screen across all human genes using this CRISPR-Cas9 technology to identify new genes which we can target or knock out, and as they go down, the frataxin protein will go up. And this, we think, will be a new method to identify new targets suitable to treat Friedrichs Ataxia. Thank you.